Good morning. Can you hear me? Good morning, colleagues. Let's start the WHO information on the compositional influenza virus vaccines for use in Southern Hemisphere 2023 influenza season. I'm Wenqing Zhang, the head of the WHO's Global Influenza Program in WHO headquarters, Geneva. I have the pleasure to introduce our director, Dr. Sylvie Briand, the director of the Department of the Epidemic and Pandemic Preparedness and Prevention in the WHO headquarters, Geneva, to be here with us to open the meeting and welcome everyone. Sylvie, please.
Good morning. It's an honor and a pleasure to join you today to open this very important meeting and to welcome you in Dublin. I first want to thank you for your tireless and continuous efforts to support influenza preparedness and response. This year, we celebrate the 70th anniversary of GISRES. Acknowledgement goes to all members in GISRES in 158 institutions hosted in 127 countries, areas, and territories. Acknowledgement also goes to countries hosting GISRES and all the partners, including you. You all together have made GISRES a true global asset for health security. After a period of low influenza activity during COVID-19 pandemic, seasonal influenza epidemics resumed. And we know that with sporadic but continuous human infection of avian and swine influenza viruses throughout the COVID-19 pandemic, an influenza pandemic is a real threat and could occur at any time. GISRES has been the backbone of influenza preparedness and response, but we are so fortunate that the network is agile and nimble, thus allowing it to support so swiftly and gravely the COVID-19 response, while continuing, of course, monitoring influenza circulation around the world. The successfully demonstrated influenza vaccine virus selection process for influenza has contributed also significantly to the deliberation of the COVID-19 virus vaccine selection process. The co-chair of the TAG-COVAC, the committee in charge of doing this vaccine strain selection for COVID-19, the director and the co-chair of this group, Kanta Subaru and David Wentworth, are also director of the WHO CCs of GISRES. Member states are looking at us to define sustainable management of SARS-CoV-2, and the use of existing sentinel surveillance will be critical for simultaneously monitoring influenza, SARS-CoV-2 virus, and other respiratory virus with epidemic and pandemic potential, as well as supporting our public health objectives. With your support, WHO has developed various guidance and resources for how to leverage influenza sentinel surveillance to monitor SARS-CoV-2, but now we must define the strategic direction of an integrated disease surveillance approach. GISRES, as demonstrating every day, will be at the center of this new global surveillance system for respiratory viruses. In the past 4 days, a WHO consultation took place here in Dublin. It was chaired by Dr. Hideki Asekawa from Japan and NIAID and Dr. David Wentworth from US CDC. The expert reviewed and analyzed massive volume of data from GISRES surveillance and other studies, and recommended viruses for use in the Southern Hemisphere 2023 influenza season. So thank you to all WHO CCs and essential regulatory laboratories of GISRES, the OFLU network, and the academia groups participating in the consultation. Thank you to all national influenza centers of GISRES for their hard work in countries to make the update of vaccine composition possible. I would also like to thank influenza vaccine manufacturers for the support to public health and the contribution to the partnership contribution under PIP framework. This contribution is instrumental to strengthen GISRES and influenza control and surveillance at large. My last thank you will be for John Macaulay, who has been here advising us for many years. And also we know that this officially is his last influenza vaccine composition meeting. Officially, I said, because usually when people retire, they continue to support WHO. So I hope he will do so. And but still, I would like to thank him heartfully for his contribution and all the support he has provided to WHO and its member states for the past years. So with the influenza epidemic back in the context of circulating SARS-CoV-2 virus, it's even more important to get prepared for an unusual influenza season. And influenza vaccine with the updated composition are an effective way to save lives, especially for people who are more at risk of severe disease. So I wish you the best for another smooth vaccine cycle, and I wish you a very successful meeting today. Thank you.
Thank you very much, Sylvie. And I would like to also add my welcome to everyone who presents here in Dublin, and also who connected remotely now via Zoom, and maybe also people connected at the moment or viewing the YouTube live, and anyone that will be view these videos that will stay in YouTube. So welcome everybody. Let's proceed to the next agenda item. As you all know that this information meeting of influenza vaccine composition and a round-the-table discussion is always chaired by an advisor. This time we discussed with the group of advisors, and we've invited Dr. Paul Benford from the WHO Essential Regulatory Laboratory in TGA, Canberra, Australia, to be the chair of this information meeting. And Dr. Benford has kindly agreed. Let me ask the floor if there's any objection against Dr. Benford to be the chair of this information meeting. Hearing now. So with this, I just hand over to our chair, Pearl, please.
Thank you very much, Wen-Ting. I too would like to express a warm welcome to everybody here in the room and those people that have joined online. I know it's getting late for many, so it is very nice to have you all joining today. It is absolute my pleasure to be here and see many of you face to face. I've been joining the WHO consultation for over 2 years, but it's the first time that I've been able to join this consultation face to face. So it has been my pleasure. I'm not going to labour the point too much because I know that you're all here to have a look at the data and have those presentations. So I might just kick on straight through. But before I do, I will kindly ask everybody, for the advisors, to introduce themselves, starting on my right.
Good morning. This is Hideki Hasegawa from WHOCC at NIAID Tokyo, Japan.
Good morning. I'm Kanta Subaro from the WHOCC in Melbourne.
Good morning. Dave Wentworth. I'm from the WHOCC at the CDC in Atlanta, Georgia.
Good morning. This is Ping Yi from WHO Essential Regulatory Labs in Food and Drug Administration in USA.
Good morning, I'm Richard Webby from St. Jude Children's Research Hospital, Memphis.
Good morning, John Macaulay from the WHO CC and the Francis Crick Institute in London.
Good morning, everyone. My name is Otto Englert. I'm from the WHO ERL at MHRA in the UK. You will know us as NRBSC, but we've recently changed our name, so we're now known as MHRA.
Wonderful. And I know that we've got two advisors online, so we might open the mics for those advisors online to introduce themselves.
Hello, this is Dayan Wang from WCC Beijing, China.
Good morning, this is Elena Gavrilova, WHOCC Vector in Russia.
Wonderful. And as Wen Qing introduced, I'm Pearl Bamford from the Central Regulatory Laboratory in Australia. So we will kick on with the introduction, with the presentations following these introductions, and I would kindly welcome John Macaulay to the podium to go through the H1N1 data.
Okay, thanks very much, Pearl. How do we get the slides? There we go. Okay, well, actually, that's not right. So we should have, the first set should be the summary of influenza activity. Christian? Do you think, okay, look, let me do it like this then. All right, okay, so it's just the wrong title. Okay, I'm not going to do that. I'm going to do, sorry, this is going to be more complicated. No, that's fine. We can run with it. Thank you very much. Okay, so what I will come on to next after this is the summary of the H1N1 activity. Okay, but I'm going to start with the summary of the influenza activity. So this is the number, the red line there is 2022, the number of viruses processed by GISRIS by week across the year. The drop-off there, I think, is, so the red is this year, and I think the drop-off is solely just an artifact of reporting, and that will probably get filled up later on, and so the numbers won't drop down like that. So overall, then we can see that over the last, so we actually analysed more samples, for example, than the last time there was a proper flu year in 2019. Why that's the case? Presumably there's just more testing going on within the system. But let's go on. This is the overall activity from the last time we had a proper influenza season of January 2020. And so what you can see here, so we're going to January 2022 through to, or end of 2020 through to July. What you can see there is the double peak of this last season. We've missed 2 Southern Hemisphere seasons and 1 Northern Hemisphere season over this period of coronavirus. So, you know, that's the situation, and most of those that have been subtyped have been H3N2. That's emphasized here, so the non-subtyped is there, but you can see the colored in the little pie chart there is that of those analyzed there, a quarter, sorry, it's not really a quarter of them analyzed. You can see there that H3N2 viruses have been predominant, H1N1 and B Victoria. B Yamagata that you notice there is on 0%. And this is a concern about what's happening to the B Yamagata lineage. It may be extinct, we don't know, but it's possible that it's no longer with us. So this is where we've had viruses from over this period, from February through to August, and see we've got good representation of many large parts of the world. Viruses have been shared within the GISRIS system. And here what we can look at is in the various transmission zones, if you believe there are such things as transmission zones like this, that what you can see there is that there's been the dark blue is not subtyped, but you can see largely in most places, but not all places, the H3N2 predominates there. But you can see in Southern Africa and in East Africa, there's a considerable number of H1N1 viruses in circulation as well. But North America, large parts of the Southern Hemisphere, Amazonia and South America, there've been H3N2 viruses predominating. But you can see there as well in parts of North Africa, Influenza B viruses predominated. And in China, very interesting. I mean, you got Diane Wang on the call. First of all, it was Influenza B viruses, and then it was H3N2 taking over this period. So the numbers of samples being analyzed just by H1N1, H3N2 reflects this, and Influenza B viruses. And the red bar at the end is the most recent ones. And so you can see that the H3N2 viruses predominated in those viruses analyzed. And then similar kinds of numbers of H1N1 and Influenza B viruses. So we're going to summarize now the H1N1 activity over this period. So actually there were relatively small numbers of samples, compared to 2019. But you can still see that that is not baseline. And I probably turned it off. Where have they come from? They've come from Europe. They've come from North America and Mexico and North America. They've come from sort of small number of countries in, looks like 2 from here, but in South America, Chile and Ecuador, Southern Africa, East Africa, Central Africa, and West Africa, Europe, Asia, particularly the Indian subcontinent, and also, of course, China and Japan. So this is the breakdown by who's seen what for the H1N1. And so the red again is the colors that we see. So the Vigil had done a pretty good number. We at the CRIC had done quite a number. China, very few indeed. NIID, small numbers. In Japan, being analyzed by the Collaborating Center in Japan, and CDC, relatively small numbers compared to other years. So we now got 2 summary slides now of the reactivity, and this first one is the recognition by post-infection ferret antisera raised against the currently recommended vaccine virus A Victoria 2570, the egg-propagated virus, or the cell-propagated equivalent A Wisconsin 588. And so what you see there is, and it's different in different centers. For example, CDC is a 3-to-1 good recognition by the antiserum raised against A Victoria 2570 or the Wisconsin. They're very similar numbers here, or the same numbers virtually. So that's a sort of a 3-to-1 of good recognition versus bad recognition. For us, it's the other way around. We've got viruses from different centers, and so we're basically 3-to-1 the other way. 70% were low reactors to the current antiserum raised against the current vaccine virus. Vigil, again, is dominated by viruses that are well recognized in NIID of the numbers. They're small numbers, so we probably don't need to really go into that, but everybody's seeing the same kind of pattern. The reciprocal of this is the antiserum raised against the former vaccine virus A Guangdong Baonan SWL 1536 2019. Ferret antiserum raised against that. For us, most of the viruses, 2/3 of the viruses were recognized well by that, and some were low. And for Vigil, it was the other way around. So I think what we're looking at here is basically we've still got that same dichotomy of recognition. So this is borne out genetically as well. You're aware that this is a phylogenetic tree from the University of Cambridge, and Sarah James is here who's produced this for us. You can see there, we basically, the green colors, so that it's phylogenetic tree on the left and it's sort of a tick box type of thing on the right for a color and geographical distribution, and ticked by what months they were picked up. So the last sort of 6-8 months or so are on the right-hand side of that. And so you can see that they did split into the 2 genetic groups that we saw before, and we've been seeing that since 2020, this split into 2 genetic groups. And what you can see, though, is that geographical distribution is somewhat different, dominated by, for example, in the 6B1A 5A.1, and so that's the Guangdong Maonan group, predominant in large numbers in Europe. I don't like to say dominated because it wasn't completely dominated. Some countries were one, some were the other, but overall there were more of that older group in Europe. But elsewhere, it was the 5A2 subclade that were dominant. But we've seen changes in this group that emerged probably at the end of 2019, beginning of 2020, or it was around about then. That was when we were first concerned about this. And they've changed since then, so that the vaccine virus down the bottom there in red is the Victoria 2570 and the Wisconsin 588 virus. And so you can see there highlighted on the trees, there are two groups, you can split it into two, but they all share those amino acid substitutions there that I've highlighted in the blue color, and then it splits into two. You've got additional amino acid changes. There's one that's a group there. I unfortunately can't read that, as I can see here, but I can't be bothered. I can't read it, sorry. But the point here is that there are various viruses from Norway at the top, but there's this other group here with additional amino acid sequence changes, including 137 and 142 on different branches there. And there's a large number of viruses there from South Africa. So these viruses are in the Southern Hemisphere, and they also have been on the move. So we've also seen these in Europe as well. And these look like that these are on the move. So we need to focus on these two different groups because they're considerably different. And some of the numbers of the changes are at known antigenic sites in the HA. This is a HI table from Vidril from the WHOCC in Melbourne. And this shows this dichotomy. The anti-sera are raised against the former vaccine viruses, Guangdong Maonan SWL 1536, Victoria 2455, which is a cell-propagated equivalent to that, and A-Togo 881, also cell-propagated. They recognize the virus, the test viruses down the lower part, so kicking off there with Sydney, South Africa, they're recognized well, but the ones at the top, which is Sydney, Perth, Canberra, Darwin, South Africa are not recognized well. And the reciprocal is that the anti-sera raised against the VIC2570 vaccine virus in egg or cells, or the new virus, Sydney 5, recognized the other group well. So this is the same as what we saw before. And also what we have here is the human serum, post-vaccination human serum pool. And highlighted there in the red box is that these viruses there from South Africa in the 5A2 group are actually recognized less well, considerably less well, fourfold lower or 2 to 4-fold lower by that anti-serum versus the viruses that were present previously. So that's the 5A1 viruses are recognized better, but the human ones are not recognized so well. And that new group in South Africa were recognized less well. This is the antigenic cartography from Derek Smith and Sarah James from the University of Cambridge. And so this illustrates that antigenic cartography illustrates the results I've just shown you. These are from the CC at Melbourne and our results from London. And so you can see just absolutely black and white, these viruses are antigenically distinct. So we also got post-vaccination human serum studies, and we've got, there were 7 panels of post-vaccination human sera were used. We got post-vaccination for egg-propagated vaccines, that's children and the adults and elderly, and that was the component, was a Victoria 2570. And also the cell culture-based vaccine where the antigen was the Wisconsin 588 2019. So what we're looking here is the reduction in there's an unadjusted GMT ratio compared with the reference is a Wisconsin cell-propagated A Wisconsin 588. So that's put at 1. This is a log scale. And so what you can see here is that these viruses, whether or not they're A Connecticut 01 2021 cell propagated, one from Italy, FWG Trieste 147, Qatar, virus from Qatar. So they're all got their mean point there around about the 50% low. But that South Africa virus is way, way down. That is really low and recognized very poorly by the post-vaccination antiserum. And this is this group that's appeared with the substitutions at 137 and 142 and other substitutions, but they're recognized poorly in this panel. The antisera there, whether or not they're cell or egg, all showed the same kind of pattern. This kind of result was replicated, although it's not so marked, as the CDC data for human serology panels. But you can see here the A Ghana 2711 2022 virus. That's the 1, 2, 3, 4, 5, fifth one in from the left-hand side. And again, we're comparing against the cell-propagated A Wisconsin 588 2019. You can see there again in both the Australia panel, two of the panels there, that we're seeing that the midpoint is way below and the 90% confidence intervals are not even touching 50%. You know, the upper limit is way below the 50%. Although the elderly is an adjuvanted vaccine, it's not quite so bad. So but it's the adjuvanted vaccine that's recognizing that virus, the Ghana virus. And this is the one with the 137, 142 and other substitutions. So this is a summary of the whole set, and the newer 6B1A5A.2 viruses there are the ones we need to focus on, because this is the group that seems to be expanding and being particularly at the threat in the Southern Hemisphere. And what you have here is when you're in blue, it's sort of okay, recognition. But if there's an X in it, it means that you should disregard that result because the homologous titer was low. Too low to really make a valid comparison against. But the other panels, most of these other panels that you can see there against the Connecticut 01, this is done by different centers, whatever, but also we got the South Africa virus and that Ghana virus, as you can see there again, these are really low recognition by the post-vaccination antisera. So this is when I saw this initially, you just go, this is a problem. Just moving on to the antiviral resistance of the H1N1s, there was just 5 viruses that were identified as showing resistance to oseltamivir and peramivir, and 4 of which were analyzed phenotypically. And of 607 H1N1s, there was no evidence of genetic or phenotypic change associated with resistance to baloxavir marboxil. So to summarize the summary, globally relatively few viruses, H1N1s, were collected since January. They've fallen into that group, but the group splits into two, the 2 subclades within the 6B1A5A. The 5A1 group, they predominated, you can remember back to the green nature of the phylogenetic tree from University of Cambridge, they predominated in Europe. They've got amino acid substitutions at 187, 189. The other subclade, and its more general circulation around the world and has been seen in Europe in recent months, defined by additional amino acid substitutions. So that's the main part of that tree, but they've also evolved further with additional substitutions, K54, 186, 189, 224, 259, and 308, and these numbers of those are in known antigenic sites. As you've seen now for 2 years, that these 2 genetic groups are antigenically distinct. Some are recognized well by ferret antiserase against Gundongman and SWL 1536, and others are not. And conversely, the other set are recognized well by the A Victoria 2570-like virus, antisera raised against 2570-like viruses, but recognize the other ones poorly. You can see that very clearly in the antigenic cartography made by the University of Cambridge. There have been additional amino acid substitutions, and the substitutions that we're seeing, and particularly those with additional substitutions at 142 and 137, are really poorly recognized by post-vaccination antisera. And this is just a summary of what I just summarized before about the sensitivity to antivirals. And so a small proportion were reduced susceptibility to the neuraminidase inhibitor or oseltamivir, but most of the viruses were expected to be sensitive to baloxavir.
Thank you very much, John. Are there any questions from the floor for John? Any questions online? Oh, yes.
Well, I noticed on the isolation of the rates and that Stephen Rockman, Seqirus. John, I noticed on the isolation rates from the different centres of egg versus cell, the numbers were absolutely exact from egg and cell. And I found that really surprising.
Well, I mean, we're not really seeing much difference there between the two. They are pretty black and white in the way that those are reacting. That's been the case for these.
I think you might want to go back and look at those numbers.
I don't think so. I think we're quite happy with those numbers.
Any further questions from the floor? For those online? I'm hearing none. So now we will welcome Cantor to the floor to present the data for the H3N2. Thanks, Cantor.
Thank you very much. So as you heard, I'm going to summarize our discussion and deliberations about the H3N2 viruses. This is a summary of the number of H3N2 viruses detected by GISRIS. The red line represents 2022. So as you will all recall, we had virtually no influenza activity in 2020. That's the black line. A bit of a return in 2021, which was the yellow. And that sort of climbed and that's followed on in the red line there. So H3N2 viruses predominated globally. So this is a map showing you influenza A H3N2 activity. There is a glitch and Australia should be red. So we had quite a bit of influenza activity in Australia this year, predominantly H3N2, although we had some H1N1 PDM09 as well. So H3N2 viruses have diversified over the years into many different clades, and we've had 2 clades that have circulated in different parts of the world. Most of the world has seen the 3C2A1B 2A2 that I'm going to abbreviate to 2A2. So I don't trip over all the alphabets and numbers. And they predominated globally. The 3C2A1B 2A1A viruses that I'm going to call 2A, I'm going to call 2A2, the 2A1 were identified in China and earlier in the year from Timor-Leste. So most of the world saw 2A2 and there was some 2A1 activity. So this is a mega tree similar to what you saw with the H1s, again, courtesy Sarah James and Derek Smith from Cambridge. And you're looking at the geographic distribution that's color-coded. And you could see we've identified here the 2 vaccine strains from the 2022 Southern Hemisphere and the 2022-2023 Northern Hemisphere. The Darwin 6 is the cell-grown and the Darwin 9 is the egg-grown representative. So we see a lot of circulation of 3C2A1B viruses. We see very little of those other ones and a small contribution of the 2A1s and a larger contribution from the 2A2s that was scattered around the world, similar to what I showed you on the other map. Now, if we drill down and look at a phylogenetic analysis of the H3N2HA genes, this is from Vidril, and it's just to show you the dominance again of the 3C2A2 the 2A2 viruses compared to the 2A1 viruses. There has been continued diversification of the 2A2 viruses, notably amino acid residues H156Q, H156S, and changes at D53 to either an N or a G. I don't expect you to find those. So if we look at the antigenic characterization over the last 4 years, the red is from this year, and you could see, again, how much we saw in Australia of H3N2 viruses and the CRIC and CNIC as well. This is an example of a virus neutralization assay that in our hands is called a FRA assay. This is dated from the 25th of July. And it's really to show you how these are the two vaccine strains, Darwin 6 and Darwin 9. They recognize the 2A2 virus as well, whether they have the 156S change or not. And they do not recognize the 2A1 virus as well. So there's a reduction in recognition of the 2A1 viruses. And the converse is true if you use the 2021-2022 Northern Hemisphere vaccine strain, which was a 2A1 virus, Cambodia-like viruses. They recognize the 2A1 virus as well, but not the 2A2 viruses. So we've seen this pattern before. Testing using human serum pool from Australian adults who were vaccinated with the Darwin 6 or with the Darwin 9 egg-containing vaccine, we see that they recognize viruses from both 2A1 and 2A2 well. There's a bit of a reduction in a group of viruses represented by some viruses from South Africa. So now if we summarize the data from all of the CCs that have tested H3N2 viruses in HI assays, looking on the left are the reactivity against the Darwin 6 cell-grown virus, which is a 2A2, and the Darwin 9 egg-grown virus on the right. And we see that 92% of the viruses tested reacted well with ferret antisera raised against the Darwin 6 cell virus. And it was about a 50-50 split when you look at ferret antisera raised against the egg-grown Darwin 9 virus. Now, if you go back a bit and look at the ferret antisera raised against the Cambodia 2A1 virus, the order is flipped there. 78% were low reactors to the Cambodia virus, which is consistent with the fact that we've evaluated a lot of 2A2 viruses in circulation. And the ratios are very similar between both the cell and the egg-grown Cambodia virus. Now, we also use virus neutralization assays in the CCs, and the data looking at the reactivity against ferret antisera raised against the cell-grown Darwin 6. 98% of the viruses reacted well with the ferret antisera raised against Darwin 6, and about 62% reacted well with the ferret antisera raised against the egg-grown Darwin 9-like virus. And if we flip back again to the Cambodia strain, it's about a 50-50 split when we test against ferret antisera raised against the 2A1 virus, and 86% were low compared to when tested using ferret antisera raised against the Cambodia egg-grown virus. Looking at antigenic cartography, and these are data and figures supplied by Sarah James and Derek Smith, the H3N2 hint assay from the Collaborating Center in Atlanta on the left, HI data from the Crick Institute in the middle, and HI data from the CC in Melbourne on the right. You can see that the Cambodia virus and the 2A1 and the 2A2 viruses are quite antigenically distinct on all of these figures. And the viruses that have circulated largely in this past season cluster well around the Darwin 6 and Darwin 9-like viruses. Analysis of the post-vaccination human sera. These are from adults who received, adults under 65 and elderly over 65, that received the Darwin 9 and Darwin's 9-like virus, tested against a variety of 2A1 and 2A2 viruses, when we compare the GMT virus neutralization titers on this slide against the GMTs against the Darwin 6 cell-grown viruses, all of the sera tested against a variety of viruses gave us GMTs that were not significantly different. So to summarize, influenza A, H3N2 viruses predominated globally. In terms of phylogenetics, we have an ongoing evolution of these viruses. They fall into two clades that we call 2A2, abbreviated to 2A2 and 2A1. And I've listed the shared amino acid, shared HA1 substitutions in the 2A2 viruses, but they have further diversified into genetic groups that typically encode these changes at 156 and 53. The 2A2 viruses are defined by a group of shared HA substitutions at 186, 193, 195, and 198. They predominated in China, and some of the viruses from China have additional substitutions at 171 and 48. Antigenically, the 2A2 and 2A1 viruses are distinct. The ferret antisera raised against the Darwin 6, which was the Southern Hemisphere 2022 cell recommendation, and the Darwin 9, which is the egg recommendation, recognize 2A2 viruses with the H156S residue well. They reacted less well with 2A2 viruses without the H156S substitution and reacted poorly with the 2A1 viruses. In contrast, ferret antisera to the 2A1 virus, which was represented by the Cambodia 2020 strain, reacted well with the 2A1 viruses, but less well or poorly with the 2A2s. The human serology data using serum panels from people vaccinated with the Darwin 9 2A2 virus in the vaccine, the geometric mean HI and virus neutralization titers against recent representative 2A2 and 2A1 viruses were not significantly reduced compared to titers against cell culture propagated Darwin 6. Reductions in virus neutralization GMTs were more pronounced when compared to the egg-propagated Darwin 9-like reference viruses. In terms of antigenic susceptibility, 4,577 H3N2 viruses collected since February 1st, 2022 were tested by genotypic and phenotypic testing, and none showed reduced inhibition to neuraminidase inhibitors. And of 3,250 H3N2 viruses and tested for susceptibility to baloxavir, none showed evidence of genetic or phenotypic reduced susceptibility. And so with that, I will thank you and hand it back to Pearl.
Thank you very much, Cantor. Any questions for Cantor on the floor? Any questions online? I'm hearing none. So without further ado, we might move on to the bees, and I will welcome David to the floor.
Thanks very much, Pearl. All right, so I am going to move through the bees for you. Yeah. So here we're looking at the global circulation of influenza B viruses and looking at multiple years. You can see from 2018 through 2022 shown from left to right here. And you can see that 2018 was really the last time we had a large B Yamagata epidemic. And the viruses, the lineage is determined by have color coding. So B Victoria is green, B Yamagata is blue, and those that are undetermined are orange. And so that you can see that the B Victoria lineage viruses have really dominated the epidemics more recently. Here I'm showing you the viruses detected, the B viruses detected by GISRIS. And again, we can just focus on the 2021 in the yellow as it climbs through the year and then becomes into the 2022 into the red. So you're looking from the right-hand side of the slide and coming into the left-hand side, and as it's declining now and more flat in this period. This slide shows you the lineages as their percentages determined. And so what you can see is for the viruses where lineages were determined, 79% of them were well, all the viruses where lineages determined were B. Vic. So 79% of the viruses tested were B. Victoria, 21% were not determined, and there was 0% B. Yamagata in this reporting period from February to August 2022. This slide is very similar to what you've seen by Kanta just a minute ago. This is the B virus activity now, where the increasing color towards orange is increasing activity globally. And so it just gives you a good sense of where and in which countries the virus activity was happening. And so, as was already mentioned, we saw a lot of influenza B activity in Asia, and particularly China, and in parts of Africa like Egypt. And then you can see the lighter yellow color with about 0 to 5% positive in many other countries around the world. So we'll get into the details of the B. Victoria lineage viruses next. Again, this is a large phylogeny illustrating the time course of these viruses that have circulated. And on the far right is the major clade names for the viruses that have circulated. And again, we want to thank Derek Smith and Sarah James for these phylogenies. And so what you can see is, in 2020, we had a large split of viruses into the 1A3, which contained a triple deletion of 162 through 164. And so you can see that on the far left side, all the tick marks towards the bottom of that tree, multiple countries where those viruses existed. But then there was a big fall off in the SARS bottleneck, basically. And then we had them reemerge in a subgroup of the 1A3, which was named 1A3.A. These contained N150K and the number of substitutions listed there. And you can see that these primarily circulated in China. They split into two groups, pardon me. The 3A1, which really only circulated in China for the most part, and the 3A2, which started to diversify and circulate globally. So you can see that by the color coding and the tick marks towards the right of that tree in the 2022 columns. Now I'm going to give you a little bit more detailed tree. Actually, I should point out the B Austria 1359417 2021 virus is in the base of that major 1A3A branch of the tree. So now we're going to look a little closer at a smaller set of viruses to kind of point out some key viruses that we'll talk about later. So again, now this tree is moving from in the evolutionary sense from the bottom of the tree towards the top. And what you can see is the 1A3 viruses, we've still had a little bit of circulation that was pretty hard to see on the tree before, but there were a few tick marks and they were green towards in 2022. And so those are highlighted in the green dashes there that occurred in April and May and June. And so this is just viruses that have continued to evolve from the base 1A3. They've acquired additional substitutions, which are boxed in red there, the K75E, E128K, T155A. And I'll point out the 155A on another slide, but it represents that whole group. A virus representing that group is the from Kenya there, and it will appear in our serology assays. And then the older viruses like B. Washington are above that in the red. And then you see B. Sichuan Jingyang as a 3A1 virus. And then further up, now you get into that large group of viruses that are all 3A2 viruses. And so the next group that is boxed is that's the defining set of amino acids in the 3A2 viruses, which are the 127T, 144L, and K203R. Right above that box, you'll see H122Q, and that's represented by the B. Henyan Jingyang 1118 virus. And those primarily circulated or almost solely circulated in China. So they just added one additional substitution. We've also seen changes a little bit higher up. They're represented by the B. Maryland virus, such as the T1822 and D197E. We have another group, and then just above that, you can see the B. Austria, the current vaccine virus, and another group boxed in red there, the D197E group, represented by like the B. Connecticut or Massachusetts viruses there. All right, so this slide illustrates the clade diversity in two different time frames. So the collection dates from September 1st, 2021 to January 31, 2022 on the left, and from February 1, so this period, to August 31, 2022 on the right. And the main point of this is so that you can see in different countries some of the different clade circulation, but also so that you can kind of appreciate a decrease in the 3A1 viruses and an increase in the 3A2 viruses. And you can particularly see that in China, where it represented about a quarter of the viruses there in the last period and now represents less than half of that. It has continued to decline there. This is a little bit easier way to see the clade turnover here. On the far left are the older viruses where there was quite a bit of 3A1 and 3A still circulating, and the 3A2 is still the predominant group. But over time, the 3A2 has continued to increase and the 3A1 has continued to decrease, with the base 3A still hanging on. As I pointed out, a few of those viruses are circulating. Here we're looking at the viruses that were antigenically characterized in the last 4 reporting periods, and the most recent period being in the purple. And it's quite easy to appreciate that the CNIC saw a lot of influenza B viruses and had many to antigenically characterize. Francis Crick Institute also characterized quite a number of influenza B viruses, and the rest of us didn't have that many around to characterize. So this summarizes the data with B Victoria antigenic analysis with just 2 sera, the sera against the Southern Hemisphere 2022 vaccine antigens. On the left are the B Austria 1359417 2021 cell antigen representative. And you can see there of over 1,000 tested, 92% are antigenically like that cell antigen. And the same number, 92% or proportion, is recognized by the egg antigen. So that's a very good result there. And here you can clearly see the antigenic distinguishing characteristics. Again, this is cartography of the HI data that's provided to Cambridge. And you can see the older viruses like B on the left-hand side. We can focus on the CC Beijing HI data. On the left-hand side, you can see the older viruses like the B Washington previous vaccine virus is in one position. And each of these squares represents about a 2-fold difference in an HI test. So those light squares are telling you how far away these different groups are. And the color coding illustrates that most of the viruses in circulation, the 3A2 viruses, are up in the upper left-hand corner there, and you can see where B Austria egg and cell antigens sit. And then you can see in this little bit darker green color there, the 122Q virus is tested. And so they're all interspersed in there. And so that 122Q is really not impacting antigenicity as we can detect here. And the 3A1s are the dark green viruses down in the left-hand corner. Now in the CDC data on the right, you can see the same kind of pattern. We don't have any of the 3A1 viruses, so you don't see that dark green bunch of viruses that you see in the CNIC data. But one thing that I didn't want to show you here was if you look at the B Washington data, we still see the exact same pattern as CNIC does. But we have these 3 purple dots that you can see there. Those are those very small number of viruses with the T155A substitution. So they're the older 1A3 clade viruses. And you can see they are related to the Washington-like viruses, the older viruses that are yellow, but they are pushing further away. And so they're showing some antigenic distinction as well. Again, those were circulating in very few numbers. This slide illustrates a serum circle. So what it's showing you is within a 4-fold of homologous titers, all the different viruses that would be neutralized well by the antisera to B Austria cell-like virus or egg-like virus on the left and right. And so that's good. This slide drills into the data a bit more. I know it's difficult to read these HI tables. This is one from CNIC, who's the group that saw so many influenza B viruses. And what you can see is the older viruses are on the left-hand side, and so the color coding helps a bit. But we have good reactivity with the 1A3-like viruses, but really only the B Washington there is tested. Now the 3A1 group is highlighted in the light blue at the top. And you can see, again, good reactivity with that group, and there was only 1 test antigen in this test because they're becoming so few. It was a pretty recent test. And you can see that that was covered very well. And then you get this split with the green boxes of all the 3A2 viruses. And even those with the 122Q are being neutralized pretty well, or very well, by the Austria 1359417 egg and cell antigens, or antisera to those antigens. This slide summarizes a lot of human serological analysis. And I know it's kind of small to read, but the main take home, and John's already introduced this, is the blue is really pretty good recognition. We're not too worried about it. And as you get into the darker orange, that's when there's potential non-inferiority, or there's potential inferiority for the antigen. And so I think you can really appreciate, just looking at the top headers, the big groups, the 3A2 versus the 1A3 and the 3A1. So the 3A2 all being covered very well, including the Henyang 1118, which has the 122Q substitution, all the viruses that have the D197E substitution. That's the Massachusetts 01-like virus. Additional one with a lot of changes, which the 122Q and some insertions also covered fairly well. And then the Maryland 1 covered very well, which had the T182A and D197E. So you can see that 197 is being evolved in a parallel evolution pattern. Now on the 1A3, what you can see is some poor reactivity there, and those are some of the more recent viruses that I pointed out, like in Kenya, and there's another representative from the Netherlands where those viruses have been circulating. And then, so those are derivatives of the older 1A3 lineage, the base lineage. And then the 3A1 viruses that have been on the decline are on the far right. So to summarize the NA inhibitor activity, 858 viruses were analyzed. One had amino substitution D197N in the neuraminidase with reduced susceptibility to neuraminidase inhibitors. And the endonuclease inhibitors, 497 B Victoria lineage viruses collected in this period were tested, and none showed evidence of reduced susceptibility to baloxavir, by sequencing of the PA gene. So for B Yamagata lineage viruses, John's already touched upon this. There have been no confirmed detections of circulating B Yamagata 1988 lineage viruses after March 2020. And so as a result, no B Yamagata lineage viruses have been available for analysis during this period. So to summarize all of what I just told you about the influenza B virus, only B Victoria viruses were available for analysis. When it comes to the hemagglutinin phylogenetics of the B Victoria lineage, all are derived from the 1A3 clade that has a deletion in the 162 to 164, the so-called triple deletion variant from quite a long time ago now, and the K136E substitution. A small number of viruses derived from this group continue to circulate, and the recent viruses that were from Kenya and the Netherlands have evolved these additional substitutions, which I won't read out to you. You can see them there on the slide, and I've discussed them already. And the subclade 1A, 3A viruses with HA genes that have these other substitutions, 150K, G184E, N197D, and R279K have predominated. And as I showed you in the evolutionary trees, both the big and the small, they've split into two groups, the 3A1 and 3A2. 3A1s have the B220M and the proline 241Q substitution. And they were seen exclusively in China and have been decreasing in number. And the 3A2 continue to circulate and increase. They are represented by amino acid changes at 127T, P144L, and lysine 203 arginine. And they've seen in Asia, Africa, Oceania, Europe, North America, and South America. So they're really just globally distributed. And the majority of these viruses now have the D197E substitution in the HA1. So to look at their antigenic characteristics, post-infection ferret antisera raised against the B Austria 1359417 2021 like viruses, which is a 3A2 virus, inhibited the majority of recently circulating viruses well, which are mostly 3A2 viruses, but recognized the 3A1 viruses poorly. The subgroup 3A1 and 3A2 are clearly antigenically distinct. That's very easily to appreciate from the cartography I showed you. And a small number of these 1A3 viruses that were detected in Kenya and Netherlands were not recognized well by ferret antisera to the B. Washington viruses. Those were those purple dots on the cartography I showed you, and were poorly recognized by antisera to the B. Austria antigen. In human serology studies, I just wanted to remind you that that vaccine contained the B. Austria antigen that was given to humans. And the majority of recent representative B. Victoria lineage viruses from the 3A2 subgroup were well inhibited by that sera. And that was all the blue you saw on that large summary slide. And the significant reductions in GMTs were detected in most serum panels for the viruses from the clade 1A3 that had those additional substitutions, the K75E, E128K, and T155A, as well as the G230N. With regard to antiviral susceptibility, one virus had an NA amino acid substitution at D of D197N, and that's associated with reduced susceptibility to neuraminidase inhibitors. And all the viruses analyzed showed normal susceptibility to neuraminidase inhibitors and endonuclease inhibitor with all the other ones. That concludes my part.
Thanks, Dave. So we'll open up the floor to any questions for Dave. Any questions online? Okay, with no questions, we might move right along. And as you know, that we don't just analyse the seasonal data, we also do some zoonotic analysis as well. So I'll open up the floor now to Richard, who will present the zoonotic.
Thank you, Paul, and hi all. Yeah, so as Paul said, and as you all know, we spend a considerable amount of time on the first day of the deliberations summarising what's been going on in the animal-human interface. And so to look at this, you know, this is just a number of human infections with animals from human infections with viruses from other animals and birds over the past few years. And as you can see, we've had an increase in the number of reports of these and the diversity of reports of these over the past few years. And this also includes the period since the last time we met in February. So there have been 27 zoonotic influenza A infections reported over this time. We're going to go through most of these and very briefly. Because of time, I'm not going to touch upon the last two there. There has been one H10N3 infection reported in China. This was a wholly avian virus and someone who had exposure to poultry. And as you can see, in this period, we have had no AH7N9 infections. But quickly run through these. So the influenza H5s. What we do know, of course, is that these are viruses that have been in birds for some time now, but over the past couple of years, we really have seen an explosion in activity of this virus within bird species. And if we look at over the last few months, we really have seen continued spread of these 2344B viruses over most of the world. These are in the form of H5N1 and parts of Southeast Asia, including China, often associated with the N6 neuraminidase. But we have seen this virus, and again, I think the notables are, as you sort of highlighted by the number of dots here, the sort of establishment or potential establishment of these viruses in Europe, in North America, and down into Africa as well. We'll look at the flip side of this in terms of how many of these viruses have actually been detected in humans. So even though I think the dominance in the birds is the H5N1 in terms of what's being detected in humans, it's actually the H5N6, so 7 reported cases from China. There's all with poultry exposure and 1 case in the US. This was an individual who was actually involved in depopulation of an infected poultry premise. So again, close human infected bird exposures in all these cases. Not all of these viruses could be isolated, but certainly genetically, they look similar to available candidate vaccine viruses. And you can see at the bottom there where we are with the cumulative numbers of human infections with these goose guandong lineage viruses. if we look a little bit, drill a little bit more down into the genetics of these viruses, particularly, this is including those in animal populations and birds. So this is from our OFLU colleagues, showing that we're now starting to see a little bit of geographic clustering of these viruses. That hasn't necessarily been the case over the past few months, well, prior to this period, the past few months, but now we're starting to see it fall into this. So you'll note the lineages in North America. Also want to draw your attention to that sort of evolving group of viruses from Africa and the yellow there. And if we look antigenically, up until this point, the A-Astrakhan CVV has been actually very, very effective in terms of inducing ferret antiserum that cross-reacts against all these 2, 3, 4, 4Bs. But in this period, and again, this is data from OFLU colleagues, we've started to see a drop in the recognition of antiserum generated to that Astrakhan CVV to some of these viruses, particularly some in Eastern Europe and those viruses through Africa. And conversely, as you can see here, a virus from Ghana here, from a chicken in Ghana, ferret antiserum generated to that, does a really good job of recognizing all of these viruses. So sort of as highlighted down the bottom there, we have recommended generation of one new CVV for the H5s, and it's a virus. We haven't got an exact virus to name now, but it'll be something antigenically like that chicken Ghana virus there. H9N2s, again, the activity both in humans and avian species with this well-documented zoonotic virus has been pretty consistent with previous periods. We've had 5 reported events in China in humans, 1 in Cambodia, or with the Y280 lineage H9 viruses, which are endemic in poultry populations in that region of the world. There have been no reports of human infections with the other major lineage, the G1, but we know that these continue to circulate in poultry populations in more in the Central Asia and Africa. And again, the one thing about the H9N2s, although they do seem to accumulate a number of substitutions in HA, their antigenic properties remain very, very stable. And so we do have pretty good coverage of all of, well, most of these emerging or new viruses with existing CVVs. Something a little bit new this time around was H3N8. So these were 2 reported human cases from China, 2 children infected with H3N8 viruses. These viruses were similar, as you would expect, to viruses circulating in poultry markets in the region, and in poultry, just highlighted here, sort of phylogenetically. These are phylogenetically and antigenically very distinct from the H3s that we see in humans or the H3s that we see in the swine populations. Colleagues at the H5 reference lab in University of Hong Kong have put some of these through ferrets, and these some of these avian viruses do transmit by the airborne route fairly efficiently in the ferret model. We do know there's probably some measured low seroprevalence in small cohorts of people to this virus as well. So due to these properties of these viruses, we have also proposed we move forward with development of an A-Henan-like virus to represent this group of viruses that we consider have some zoonotic potential. So moving on from things with wings to things with snouts. So looking at what's going on at the swine interface. So it's a very complicated picture. I think we know, again, provided here by OFLU colleagues, there's lots of diversity, genetic, antigenic diversity of these viruses. Of course, it's all H1 and H3, but depending on where you are in the world, there's lots of differences in the viruses that are circulating. Very, very complicated epidemiological, ecological situation. But as in previous periods, we do see these viruses being detected in humans. This time around, an H1N1 virus, viruses in China and Germany, and H1N2s and H3N2s in the United States, again, at these agricultural state fairs, where again, we've seen lots of transmission of viruses from infected swine to humans over the past number of years. So I think that's all I have. Thank you.
Thank you very much, Richard. So I'll open up the floor to any questions. No questions from the floor. Any questions from online participants? Okay, without further ado, I will kindly welcome Wenting to the podium to go through the recommendations.
Thanks very much, Paul. So we all know that the WHO's recommendation on influenza vaccine composition is based on the year-round work of a global network, the GISRIS, which comprises WHOCC's National Influenza Centers, Essential Regulatory Laboratories, and H5 reference laboratories, supported by hosting countries and partners, including GISED, including vaccine manufacturers like you connected here, as well as many other partners in both public and private sectors, as well as academia. So thank you very much. GISRIS this year celebrates its 70th anniversary. The network has come to today is really because of the collective efforts by countries, by dedicated people of several generations. We've said farewell to many dedicated GISRIS flu fighters. And today, as in the opening, Sylvie mentioned that we'll also say goodbye to Dr. John McCauley, who actually has been in this role in the past more than a dozen of years. He's playing a critical role in GISRIS, and of course, including also the vaccine composition recommendation function of the GISRIS. For those of you who will go to Belfast, please join us tomorrow evening at 6:30 in Queen's University in Belfast. Let me also to mention that just yesterday on the WHO's website, we published a WHO feature story on GISRIS. It's 70 years past. So in the past 4 days, a WHO consultation took place to review and analyze the massive volume of information generated from GISRIS and made a conclusion on the recommendation. This meeting was a hybrid of in-person and virtual meeting. It was chaired by Dr. Hideki Hasegawa from NIID Japan and co-chaired by Dr. David Wentworth from CDC Atlanta. There were 10 advisors on the committee. 8 of the 10 are on the committee for seasonal influenza vaccine composition. These advisors attended in their capacity as representatives of their corresponding WHOCCs and EILs. At the beginning of the meeting, the interests declared by these experts were disclosed to all meeting participants. In addition, 35 participants also participated from WHO collaborating centers, EILs, other GISRIS laboratories, and also from academia. Experts from WHO region offices and also HQ also participated in this meeting. So now let me summarize briefly the deliberation in the past 4 days. Influenza surveillance, which was disrupted during the early stage of COVID-19 pandemic, has since recovered in most countries. Since February this year, overall influenza activity remained lower than the same period pre-COVID-19 pandemic, but it has reached the highest level compared to the same periods during the pandemic. Influenza virus detections have returned to pre-COVID pandemic levels in many countries in Southern Hemisphere since February this year. Therefore, sufficient numbers of influenza viruses available for characterization in WHO CCs. Influenza A, H1N1, H3N2, and B viruses circulated in varied proportions. Influenza A viruses predominated in most countries, with the subtype AH3N2 predominated in many countries. Influenza A H1N1 predominated in some countries in Africa and in some countries in South Asia. Influenza B viruses were detected in many countries, but predominated only in a few countries in Africa and Asia. Now on the influenza A H1N1 PDM09 viruses, overall H1N1 seasonal viruses circulated in many parts of the world and predominated in some countries. Genetically, since February this year, the HA gene of circulating viruses belonged to clade 6B1A5A, or I just call 5A clade. Viruses in that clade further diverged into two main subclades, 5A1 and 5A2. Since January this year, 5A2 viruses further diversified into subgroups, emerging and circulating in parts of the world. Antigenically, ferret antisera raised against egg and cell propagated Guangdong Maonan-like virus, which is a 5A1 virus, recognized well 5A viruses, but not 5A2 viruses. Ferret antisera raised against egg and cell culture propagated A. Victoria 2019 viruses, which is a composition for the vaccine going to the Southern Hemisphere 2022 and the Northern Hemisphere 2022-2023 vaccines, recognized well 5A2 viruses, but not 5A1 viruses. A pool of sera obtained from vaccinees who had received Southern Hemisphere 2022 vaccines showed good recognition of both 5A1 and 5A2 viruses, but less well of a circulating subgroup with additional amino acid substitutions, including 142R and 137S in HA1. In human serology studies using post-vaccination human antisera, antibodies induced by A. Wisconsin 2019-like vaccine viruses showed minor reductions in post-vaccination HIGMT for the majority of recent viruses, but significantly reduction have been observed for some 5A2 viruses, including the above-mentioned emerging subgroup. Summary of the H3N2 viruses. H3N2 viruses predominated in many countries since February this year. Genetically, 3C2A1B2A2 or 2A2 viruses predominated globally and further diversified into 4 groups circulating in different proportions in different regions. 3C2A1B2A1 or 2A1 viruses predominated in China and early this year also in Timor-Leste. Antigenically, ferret antisera raised against 2A2 viruses, for example, cell culture propagated A. Darwin 6 or egg propagated A. Darwin 9 viruses, which are the composition of the vaccine in Southern Hemisphere 2022 and the upcoming Northern Hemisphere, inhibited well 2A2 viruses, but less well 2A1 viruses. Human serology studies using post-vaccination human in those studies, geometric mean titer GMTs in HI and virus neutralization assays of antibody against the majority of cell culture propagated Reagent 2A1 and 2A2 viruses were not significantly reduced compared to those against the cell culture propagated Darwin 6 vaccine virus. Now let's look at the B. Since February this year, influenza B virus detection account for 4.7% of overall virus detection by GISRIS. About the B Yamagata lineage viruses, actually after March 2020, there have been no confirmed detections of circulating B Yamagata lineage viruses. On the B Victoria lineage viruses, since February this year, nearly all viruses fell in subclade 1A3, which has a triple amino acid deletion in HA1, with the group of 1A3A group predominant. This group virus 3A further diversified into two main subgroups, 3A1 and 3A2. 3A1 viruses represented a small proportion and only detected in China in early 2020. 3A2 viruses detected in Africa, Asia, which also includes China, in Europe, North America, Oceania, and South America. Ferret antisera raised against B Austria 2021-like viruses, the vaccine composition in Southern Hemisphere and upcoming Northern Hemisphere inhibited well this subgroup 3A2, but not 3A1 viruses. B Washington 2019-like virus, which is a Northern Hemisphere 2021-2022 vaccine composition, and the B Austria 2021-like viruses recognized poorly 1A3 viruses that continued evolving. Human serology studies did not show significant reduction in post-vaccination anti-hemagglutinin antibody GMT against the majority of recent 2A2 viruses, but more pronounced reduction observed against 1A3 viruses compared to those of cell culture propagated B Austria 2021 vaccine viruses. So based on this analysis, it is recommended that for use in 2023 Southern Hemisphere influenza season, vaccines of quadrivalent egg-based contained A Sydney 5 2021 for H1N1 component, A Darwin 9 2021, B Austria 1359417 2021, and a bouquet virus. For the quadrivalent cell or recombinant-based vaccines, the same component, but for the H3, there's a different format. It's A Darwin 6 instead. For the trivalent vaccines, it is recommended to include the components of H1N1, H3N2, and BVIC. So the technical report based on which this recommendation was made will be published on the WHO's website. Accompany that report, there will also be an FAQ, and also the corresponding CVVs as well as reagents will also be updated on the WHO's website. In addition, during these consultations, the zoonotic influenza virus was also reviewed, and a technical report of the zoonotic influenza as well as the recommended new CVBs for pandemic preparedness purposes will also be published on the WHO's website. With this, I stop here and back to the chair.
Thank you very much, Wen-Qing. Any questions from the floor? None? Any questions for those online? One online?
Yeah, hi.
Please introduce yourself and go forth.
Yeah, hi, I'm Suman. I'm from India, Minvex Private Limited. So my question was that since the H1N1 virus has changed for this year's recommendation, and it takes some times for the anti-sera to be generated in the, like, whether in NIBSC or the IRR. So is it possible that we can receive this virus? So that, like, is there any scope of receiving this virus so that we can work on it?
I think you're just ahead of the program at the moment. We're just going to be going through the availability of CVVs and the potency reference reagents. So hold your thought and question till the next presentation. And if you have further questions, we'll address it after that presentation. Is that okay?
Yes, that's fine. Thank you.
Fantastic. Okay, so with no further ado, we'll move on to the next presentation, which I just described as being the availability of the CVVs and the potency reference reagents. And this will be presented by Zhiping.
Good morning. Right now, I think we officially passed the baton from WHO to the CCs, to the manufacturers and regional or national regulatory agencies. It doesn't mean we stop running. We still run with you until we get those vaccine on the market. So here this shows the candidate vaccine viruses. I think that these viruses can be used directly as a starting material to propagate it in cells or in eggs, then further process it as antigen, as vaccine antigens. And also, this information can be directly used, genetic information can be used to engineering life attenuated viruses or use the sequence for the recombinant protein. And then this slide shows the CVV, the candidate vaccine viruses, egg-based, because we have an egg-based, we have a cell-based, and also we have a recombinant vaccine viruses, vaccines. So this table from left to right shows that the parent viruses or prototype viruses. In this particular case, for H1N1, since we only have Sydney 5, and that is the viruses that we used to begin with this season. And this can be added to it because if depend upon if the like viruses we found it, and that can be added to it. And then the next column shows the candidate vaccine viruses. Either you can use wild type viruses or you can use reassortant. The reason to use the reassortant is that some of the wild type viruses wouldn't grow too well in eggs or cells. So the viruses can co-infect with high growth viruses and select those adequate for the vaccine usage. The next shows that what kind of a method or technique used for reassortant, there's high yield or high growth viruses. Then the column next to it is who produced this. And then the last one shows availability of where you can get these viruses. And I will show you in the end of the slide that where you can get it. And the thing is, this, as I said, this is a continued work, and then this can be updated, day by day. again, this shows the cell culture based vaccines, and similar to the what I showed in eggs. And all those information you can find in the WHO website. And I believe this slide probably will be shared with you as well. And for H3N2, since this is the same vaccine viruses are being used for the current season, then you can continue to use it. And I'm not going to go through this, but too many of them. And this is an egg-based, and here it shows cell-based vaccine viruses. Again, because we have two components for the B viruses. The one is Victoria, and here is the Victoria cell egg-based viruses. Then this shows cell-based. And then for other lineage of the Yamagata one, and again, this we have been used for many years, and this is what we have. And again, this shows the cell-based. Then for the potency test reagents, for live attenuated, you may not need to it because that depends upon the infectivity. And for the protein-based vaccines, you need to have standards to make sure the vaccine final components, are standardized, and especially if you use one set of the reagents and everybody produce vaccine, they were comparable of the potency antigens in their final product. Okay, because this reagents composed two elements. So one's the reference antigen. Usually this is relatively easy to produce, and then just for the viruses, you just grow the viruses and then lyophilize it. And antiserum you have to produce from sheep, because for the antigens to immunize the sheep, they have to be specific to HA. So that takes time to purify. That takes usually, depends to 1 to 2 weeks if you're lucky, and then immunize sheep, and then after 2 weeks or 3 weeks, and then take the serum just to make sure if they have enough titer. If titers are not good, then you have to boost it, and this takes time. And even you produce this, and then because the vaccine released from different countries, then the ARLs have to calibrate it to make sure everybody used the same set of the reagents and give you a similar results. So make sure the final product adequate. And okay, so for the Sydney, because this we just recommend it. So we have to start from scratch. So this usually for the sheep serum, it takes at least several weeks if you're lucky, or months. So I think ERLs we work our head off to make sure that it will be available as soon as possible. And also I think after this meeting, we do have the following meetings just to make sure what are the status is. So you guys, for those who are involved in these meetings, so you can get live progress on this region. As I said, because of H3N2, there's the current one, and we do have this reference antigens and sheep serum ready, and then you can continue to use it and depend upon your platform. And this shows the reagents for cell-based H3N2s, the current being used for manufacturing of the vaccines. And then this shows Victoria egg-based. Am I following flash too fast? But anyway, you can get that information. And then this shows the Victoria cell-based reagents. And then the next show the Yamagata egg-based reagents. And Then the last one shows the cell-based, by the way, the recombinant protein have, depend upon the local agencies, you may have to use the recombinant antigens to standardize your vaccines. And as I said, all those information will be in the website, and you probably will change it very quickly as well. So all the availability of the recommended CVVs and the reagents you can find in WHO website. And the same, similar to the zoonotic vaccine. And the last one shows the website that you can get it, get out of all those informations. And I hope my presentation already answered the question online.
Thank you very much, Ziping. So I might go online first to make sure that there's no further questions before opening up to the floor. No questions online? Any questions in the room? No, fantastic. So following on from Ziping, I think that yes, officially the baton is handed over to industry, but work also starts for the ARLs as well to make sure that those potency reference reagents are available to you as quickly as possible. So I know that there's a lot of work now that we're all going to go home to start to commence, but we're up for the challenge. So I think that concludes the first half of this meeting. We will break now for coffee and tea, and we'll be coming back at noon, so 12 o'clock. So for those that are online, that's about 25 minutes from now. Thank you.